We're talking about culture transfer techniques and smear prep for staining. The most important thing is to keep safety in mind, making sure that you keep your work area disinfected. So always spray down your work area before when you sit down in the lab. Of course, remember, because you just assume the person was here before you did a bad job and they didn't decontaminate everything. Make sure you guys also do it at the end of lab too. It's very important. These are part of aseptic techniques, which we'll talk more about and harp on kind of throughout the rest of the semester. So keep those in mind. Also, do not carry any glass or plastic test tubes by hand. Always make sure you guys put them in the racks because we don't want them to drop or fall. Also, make sure not to use any broken slides. If a slide breaks, make sure you guys let your TA know right away so we can handle it together. Notify your TA of any spills that happen. If they do, just let us know. It's not a big deal. Just make sure you tell us about them. Do not handle the back piece incinerators, especially around the metal parts. They are very hot. I believe they get up to about 442 degrees Celsius, something like that. So 400 degrees Celsius. It's hot enough to melt metal, so just don't stick your finger in there, okay? Don't touch it, don't grab it, it'll be fine. Goggles, gloves, lab coat should be worn at all time when you're in lab, so make sure you have them on. Also, we will be using BSL-2 agents, so make sure you guys treat them with extra care and handle like we always do. Primary objectives, just to make sure you guys know that we're trying to transfer bacteria into new culture media and how we go about doing that. Make sure you guys are prepared for that. Also keep in mind that our quiz will be covering these techniques as well as smear preparation at the end. Important thing, whenever we talk about scientific names in this lab and whenever we write them down, we always want to write them down with the proper naming convention. So whenever we're handwriting them, we would do it a specific way, but if we're writing them as a typed name, we always want to italicize them. So if we're typing it, it'd be first letter capitalized of the genus name, and then everything else lowercase in the genus and species. So the species name is also all lowercase, and it'll be in italics. If we're handwriting it though, obviously you can't write in italics as handwriting because you're not a typewriter. We then write the first letter capitalized, everything else lowercase, and then we underline the entire thing. If you do not do that, it'll be an automatic 0 for whatever name that is. So make sure you guys are very careful in doing that in, say, quizzes or exams, lab reports, etc. So whenever we talk about bacteria, we're talking about important things like nutritional needs and why we have to constantly supplement them with them. Most important one is, of course, water, which is important to all life. Also, carbon source, how we are getting our carbon to make things in our body. You can be an autotroph, so you use inorganic carbon sources like CO2 or some other inorganic carbon source. Or you could be a heterotroph, where you eat other things, so organic carbon. So we eat things that have carbon and utilize those to make our nutrition, parts of our body. We also need important things like nitrogen, sulfur, and phosphorus. Those are really important for things like energy and DNA and amino acids. Those are very important. We also have a bunch of micronutrients we need, things that we need at lower levels that aren't, they don't have to have a bunch of them, but if we don't have them, they become very difficult for us to survive. These are things like iron or potassium or iodine. All sorts of things can affect micronutrients. So they're very important, even though we only need them for a small amount. Vitamins are also very important. Some vitamins we make, some vitamins we get from other sources. There's just different ways you can get vitamins. Now, lastly is energy, how we are generating energy. Typically from our carbon source, somehow we're making our energy. You can either be a phototroph or a chemotroph. Now, if you're using energy as your sunlight, you're in that first category. If you're using an oxidative burn of chemical compounds, then you're chemotroph. So we are an example of chemotrophs. Plants are an example of phototrophs. So that's just the difference between them. So it's really easy to differentiate them. Most things that aren't plants are going to be in that chemotroph categorization. Other things that affect growth rate, aside from nutrients, which are very important, are things like temperature. So especially for bacteria, they generally fall under 3 categories, which is psychrophile, mesophile, or thermophile. If you're psychrophile, you like colder temperatures, about 4 degrees to about 25 degrees Celsius or so. Mesophile is like a nice middle temperature, so about 25 all the way up to 37, 38, up to 40. Thermophiles go from about 40 to 50 degrees Celsius, anything above basically 40 degrees Celsius. Now there's some examples of extreme ones for both. So for example, psychrophiles, there's extremely cold dwelling bacteria in Antarctica that function at that temperature just fine. There's also examples for extreme thermophiles. For example, bacteria that like to grow at thermal vents under the sea or in Yellowstone. And that's actually how we basically have all of our ability to do PCRs from bacteria called Thermophilus equus, which we got from geyser vents. So those are actually very important. So we need those extreme ones as well. So they can operate, I believe, at about 70 degrees Celsius consistently. So that's very hot. Optimal growth temperature is different than what you can function in. So optimal is like what you do best in. So humans, it's about 72 degrees Fahrenheit. We like about that room temperature, and a lot of things do. In fact, most things like about that temperature, or at least around body temperature as well. So there are some things that have a larger or wider growth range and some things that have very narrow ones. So just remember that optimal just means this is the best temperature to grow things at. pH also influences it. So if it's a very acidic or very basic environment, a lot of bacteria or even us won't grow. Like if our blood is a little outside the appropriate pH range, we just die instantly. Same thing for bacteria. They need specific ones. Some bacteria can survive very, very acidic conditions. So Heliobacter pylori, which is the causative agent for stomach ulcers, that can handle very low pHs. Oxygen availability is also very important. So it determines whether or not you need oxygen or if you're harmed by oxygen. Strict anaerobes cannot be around any oxygen whatsoever, or only very brief, small amounts of oxygen. Facultative anaerobes, on the other hand, do not need oxygen, but they are not harmed by it. So they can grow in an oxygen environment very easily. Aerobes, on the other hand, require an oxygen environment. If you take it away from them, they cannot survive at all. So most of the bacteria that we handle in this lab fall into the category of facultative anaerobe and aerobe. We do have some anaerobes that can survive, but just do really poorly in an oxygen environment. But generally they much prefer an anaerobic environment. So subculturing. What this is the process of transferring organisms from one media to another following all aseptic techniques. The primary reasons why we want to do this is to do biochemical tests, or if we want to keep a cell line growing. Actively growing cell cultures always do better than cell cultures that are like on the cusp of death and then you put them into new media. You always want to keep them actively growing because they give much better results, especially when we do tests. The rules for transferring are as follows. They're pretty simple. It depends on what media you're going into for the most part. So if we are going into something like a deep, or we're trying to make a slant, or we're trying to get growth characteristics, we're always going to use an inoculating needle, because use like a loop in a deep, it would gouge the agar too much and create a really bad streak pattern in the deep. So we never want to use a needle in, we only want to use needles in deeps. It doesn't really matter for broths because you can't hurt a broth, so loop or needle's fine. And for slants, we generally want to use a needle because we want to look at growth characteristics. But sometimes it's not important. If we're just trying to go for bulk growth, we don't care if it's a loop or a needle or slant. Inoculating loops, however, are always used in streak plates. If you use a needle on a streak plate, it would gouge through it and make a big cut. It wouldn't be very useful at all, so make sure you guys always use loops on streak plates. Broth, again, can't hurt a broth, so it doesn't matter whichever one you prefer. So how do you do a streak plate? There are lots of different methods of doing streak plates. We'll show you guys one that works really well for us. It's a very common way to separate different types of bacteria from a mixed culture. So below you can see an example of E. coli that's got a pea glow plasmid inserted into it. And so we're trying to get ones isolated that have that. So you can see that we've streaked a very heavy one into several different parts and isolated a couple of different individual colonies. Anytime we say that there's one single colony, we can say that that's probably came from one bacteria type. So it's a pure bacteria type. So we can grab that colony and be assured, for the most part, that it's just a single type of organism, not multiple ones. If we could try to grab from like a more densely populated area where there aren't isolated colonies, we can't tell that that's pure or not. So you always have to be very careful with that. We also do this a lot of times to separate out mixed cultures, since most of the things in the universe, most bacteria sit as a mixed culture with other types of bacteria. We just have to purify them out whenever we're taking it from a sample from the environment. The way we do this optimally is as follows. So we will take a lid, and if you guys see in this image here, we take the lid and we always want to lift the lid up just a little bit. We never want to take the lid all the way off because that exposes the lid and the bacteria or the plate, I should say, to all the bacteria from you breathing or just from the ventilation system, or if somebody sneezes, it's going to open up a lot of routes for contamination. So we only want to lift the lid up as little as possible for only the shortest duration of time. And when you hold the plate, you always want to hold the plate with the part that you're going to be streaking closest to the palm of the hand that is holding the plate. So if you are right-handed, you'd be holding this plate with your left hand, and that large section that has a line down the center, that section would be facing your palm. So it'd be closest to your palm. You'd lift the lid up and you take the needle with your dominant right hand and start streaking back and forth in an up and down motion, nice and slow, not gouging the agar, always dragging. So you never want to push a loop because a pushing loop will gouge the agar. If you're dragging it, you will always create a nice, smooth, even surface. And you do the zigzag pattern. Once you do this initial zigzag pattern, you'll stop what you're doing, put the lid back on, and then you'll place the loop in the Bacticinerator and let it heat up and kill off all the bacteria that are present on your loop. Once you do that, you'll take it out and let it cool for a second, And if you want to check to see if it's cool enough to touch the plate again, you'll touch it to an area where there are no bacteria, so maybe in the corner, the quadrant next to it. Touch it. If you hear a hiss, that means it's still too hot, so let it sit for a second more. If it's cool, then you can go ahead and drag across to the other lines. Now, the reason why we're dragging across is we're actually trying to lower the number of bacteria. In this first little area that we've made, the bacteria will be completely dense. There will be no isolated colonies whatsoever, so we want to drag across here and do that zigzag pattern again. We're not adding more bacteria to this plate. We're just dragging the bacteria that are already here over to the next quadrant. And this will help us get isolated individual colonies. So we'll go across 2 or 3 lines, and you want to try to avoid the tops of those arcs, because when you do the tops of the arc, then it tends to be a lot more bacteria than you're actually expecting. So try to do it kind of in the middle, like it shows in the image. You'll do the zigzag pattern. Once you do the zigzag pattern, you will put the lid back down, flame your loop again, let it cool down, touch a blank area of the auger one more time, turn the plate one more time so that the part that you're going to be swiping is facing your palm, and then streaking across 2 more lines and doing it a 3rd time. The reason why we do this 3 times, it gives us the best chance for having bacteria that are isolated colonies present on your plate. So make sure you guys follow this. The most important thing about this technique is making sure that you do not put any more bacteria on after the very first time. So the very first time you put bacteria on, that's going to be the only time you put bacteria on this plate. Every other time is just moving bacteria across the plate to lower the numbers, basically reducing it. The next media we're going to be looking at is SimDeep. So remember, we made these during Media Prep Lab. The way you do this is you're going to take bacteria on a needle. So you flame your needle to decontaminate first, grab bacteria from your stock culture, and then you will go to your SimDeep and you'll put the needle straight down, as straight as possible, and straight back up with as little wiggle as possible. If you have wiggle, it can alter the results of your media. So this is to evaluate whether or not there is motility from the bacteria. And a lot of times the way we tell whether or not there's motility is looking at there's bacteria spread throughout the entire media. Well, if you wiggle it a little bit, it'll create this triangle shape. And if you can actually see in this tube, if we turn the tube 90 degrees, it might look like it's a cone of growth of bacteria, but when you turn it 90 degrees, you'll actually see that it's just a 2-dimensional plane of bacteria because you had too much wiggle. So definitely turn your SimDeeps around to make sure that it's actually growing out from the center stab line and not just you made a mistake when you were inoculating it, which is very easy to do. So the protocol from what you guys will be doing today is you guys will get 1 stock culture organism, and each student will get 1 TSA slant, 1 SimDeep, 1 nutrient broth, and a second TSA plate. You will inoculate the first 3 with the bacteria that we give you, but the last one, your TSA plate, that will be for a mixed culture where you'll be separating them out. And this will actually be graded just as though it were a lab report. So make sure you guys save that one for last so you have a chance, like if you mess up, you can just mess up on the TSA plate one. That is not very important. It doesn't matter if you mess up on the TSA plate for your stock bacteria. It matters if you mess it up on the mixed one. So make sure you do the stock one first. Whenever you label stuff, you always want to label on the tube itself, never the cap. You'll label the organism, your name, the date, and section. You can abbreviate the organism name. So if it's Bacillus subtilis, you can write down BS or SM for Serratia marcescens, what have you. Most important thing here is always make sure you label the tube or some solid object where the bacteria are. If you label things like caps or the lid of plates, what ends up happening is I can just take the lid off of a plate. And now you don't know what that bacteria is. I can't take the bottom off of a plate. I can't take the bottom off of a tube because the label just follows around with it. So always make sure you label the tube or the bottom of a plate always. Once you do your slant, SimDeep, and nutrient broth and TSA plate for your stock organism, you will then do it for the mixed. Key thing with the mixed, a little trick, is to make sure you guys swirl it about so that it's nice and evenly distributed before you grab bacteria with your loop to do it, because otherwise they sometimes sink and separate out. So you got to make sure they're nice and spread throughout. The bacteria that we're going to be looking at today are going to be Serratia marcescens, which is our first one here. You can see it creates this nice red coloration. It also has this serrated edge to the colony, so it's got these little teeth-like edges, and you can see it's got this nice bright red coloration. Typically, this only happens at room temperature, but sometimes different variants of it can produce that at even higher temperatures. It is a motile bacteria, so when you guys look at it on a broth, it tends to be very cloudy, so we call this turbid, and it creates this nice, even cone of growth throughout the entire SimDeep because it can go throughout the entire tube. We'll also be looking at Pseudomonas aeruginosa. Now this one creates a nice growth pattern. It is also motile, but if you notice in this tube here, the TSA slant, it's got this nice green pigmentation to it, and it's generating this pigment. So the bacteria itself is not green. The bacteria is this tan color you can see on the plate, but it produces this dye called pyocyanin. It's important to remember, so make sure you guys remember that. Bacteria create these umbonate colonies typically, so they kind of look like sunny side up eggs. Because it's modal, you can see it in the sim deep. It's created this nice cone of growth, and it's facultative, so it grows throughout the entire media. For broths, it just kind of depends on how it's feeling. Sometimes it'll form a pellicle, where it will grow on the very surface of the broth because it needs oxygen. Sometimes it'll form chunks that are kind of floating or suspended in the middle because it has enough oxygen to survive. It just depends on what it feels like doing. Bacillus subtilis is next. It forms these nice, long, large colonies that are flat and kind of irregular in shape. So they got these little protrusions. We call them lobate protrusions. You can also see on the slant here, it creates this kind of arborescent look. So arbor meaning tree. So it kind of looks a little like a Christmas tree. You can see it's got these little projections that come out kind of ridgy like a pine tree. Now, how does it like to grow in broth? It just depends on what it feels like doing the stay. It typically likes to sit as sediment, so it'll sink to the bottom, but sometimes it'll be cloudy and turbid, or sometimes it'll be pellicle where it'll form these nice little chunks. It just depends. So you have to be very careful about what you're looking at. You can never just say a bacteria will always grow this way on a plate or something, because lots of factors can affect it, like how well it's doing, if it's at an optimal growth temperature, if it's too humid out. All sorts of things affect it, so make sure you guys are aware of that. Lastly is Micrococcus luteus. This one's a nice bright yellow bacteria. It is non-modal, so you can see right here it's just staying where the stab line is. It hasn't spread out any. And it's got these nice, really smooth colonies. The filiform growth on the slant is nice and smooth. There's no ridges or anything else to it. And it typically just kind of likes to sink at the bottom of broth, so you won't see much else to it. It's nothing fancy. At the end of the day, what you guys want to do is you want to do proper cleanup. The first one, if you have stock cultures, you're going to put your stock culture test tubes, either their slants or broths, on the autoclave cart. You remember, broths go on the blue rack, auger slants go on the orange rack. And you'll take anything that you inoculated and put it in your group rack and put it in the incubator on the part that belongs to your lab group. Any TSA plates that you have, you must remember to make sure to tape them up in a stack and put them in the biohazard bag. These are, of course, TSA plates that you want to get rid of. If you're growing them, tape them up in a stack and put them in the incubator. And when you put them in the incubator, you want to make sure to put them upside down. That is, lid side down. This way, if any condensation builds, it won't drip onto your bacteria because it can ruin your slants or your plate. So make sure you guys keep that in mind. Also, make sure to disinfect your work area, like we talked about last time, with Cavicide, and of course, wash your hands before you leave the lab. Now, on the second day, after we look through all this stuff, we're also going to prepare bacterial smears so we can do our gram staining the next week. Bacterial smears are actually very important, so make sure you guys do them properly. What you'll do is you guys will get a slide. You'll actually get 4 slides in total. And for every slide that you have, you'll draw a little circle on the center. The circle just kind of helps you gauge where you should be aiming on the microscope. It's not really all that important, but make sure you guys draw on the bottom as well as label it on the bottom of the slide. You want to put the bacteria on the top of the slide so they don't overlap. The way you'll do it is you'll add a small drop of water, about almost like half a drop of water. So you'll kind of squeeze the dropper bottle a little bit and then just touch the water to the slide. So you're getting half of it. That way it doesn't create too much liquid so that it'll dry better. Otherwise, because Texas is so humid, it'll take forever to dry. So this is the best way to do it, just a little bit of water. Then you'll use a sterile inoculating needle to transfer organisms to the water droplet and spread it around. You want to spread it out so it's really far throughout the entire slide, not just one area. And then you'll let it air dry. If you let it stay in kind of like just one little area and create a very blobby bit of liquid, it'll take forever to dry, and the slide won't be very good when you actually look at it after you stain it. So make sure you guys spread it out nice and evenly. Once it's completely air dried, there's no more liquid on the slide, you will heat fix it. You'll put it on the Bacticinerator about 10 to 30 seconds. It kind of just depends on how thick your slide is. I'd say about 15 seconds tends to be the best. You'll put it on the little slats that are on the top of the Bacticinerator. They're slide width, so that works really well. You'll just put them in there and hold them for about 10 to 15 seconds. And this will help heat fix them so that the bacteria will no longer be alive. Now, how do you determine a good smear versus a bad smear? Good smear, you can read text through. So once it's dry, you can read through the text. That means it's good. If it's so thick that you can't see any text, then it is bad. You don't want to see it. It shouldn't have any chunks or anything like that. It should be nice and easy to see. Now, why do we heat fix? There's 3 primary reasons. The first one is it coagulates bacterial proteins so they stick to the slide. They don't get removed easily. It also denatures enzymes to prevent autolysis, so the slide can be preserved for a really long time. Bacteria will sometimes produce autolysing enzymes, and they'll kind of blow themselves up if you don't help deactivate them when you want to preserve them. Lastly, it kills the bacteria because obviously we wouldn't want to have a biohazard just running around in your drawer. So make sure you guys always heat fix. Remember, you guys are going to be making 4 slides. So one slide will be Bacillus subtilis, one will be from Micrococcus luteus, one will be from Serratia, and one will be a mix of all 3. When you go for the mix, try not to go overkill on the bacteria you put on there. Otherwise, it'll be too thick and difficult to see. So make sure you guys aren't going like too much bacteria on the mix. Once you guys have them mixed, you guys will then heat fix them and get them prepared for next week. So after you heat fix them, you'll put them back in your blue box that you guys have, and then you'll have them ready for the next week.